Thank you, Maria. Thank you guys for coming. So let's start straight. Thanks for the introduction of myself. I should say that what I will be talking today is created by a large consortia. First mention SILAM team, which I coordinate, but also several projects which are on the introductory slide, and probably many more. Wherever possible, I try to put the references to literature which is available on the matter. Thinking about the outlook of the session, I have to confess a small ignorance part, or maybe big ignorance part. I learned about EFI just a few months ago when we were building together the proposal for cost action. So I have to say that I don't really know who is now listening to me, and usually it's very difficult for presenter. So I tried to imagine what it should be, but if some of you find things trivial or too complicated, don't hesitate to ask questions. I'll try to start from some baselines, basically setting the terms. I know that every part of environmental science speaks own language. So I'll try to say to teach you my language first, and after that we'll go into essence of the presentation. So what is the system for bioaerosols in the atmosphere, modeling, monitoring, assessment, what that means, and a little bit of applications towards the end, and then what we actually can do further on in Europe. So let me start from the claim that pollen is not the only version of bioaerosol, of course. Yes, it is affecting approximately a quarter of European population, and some countries more than that, in terms of allergy. And we are at the peak of allergic season now in most of European countries. Same in the north, by the way. We have huge peaks of birch now. But it's not the only particle in the atmosphere. We are talking about also fungal spores and bacteria and viruses, and then even going smaller, some proteins or remnants, debris of plants, and whatever else. So there is lots of them, and you see some images on that screen from the biggest to the smallest. What is also important, and that's a challenge for measurements and for model, by the way, as well, is that these guys cover a very wide range of sizes. And we start from basically a nanometer scale when we think about proteins, which are just a collection of some molecules, almost the molecule size, and all the way down to, let's say, 100 micrometers or even more than that. So, well, if we think about seeds, they can reach a millimeter size. They don't fly really far, but they still exist in the atmosphere. So there's a large variety of them. And what is also important is that it's not enough to say we have XX microgram per cubic meter, 10 micrograms per cubic meter, for instance. We have to say who these are. And that's where the big challenge actually comes to the game. Because the roles of bioaerosols in the environment very strongly depends on who they are. The role of atmosphere, if we look from another side, so it is a transport media. They don't grow in the air. They only fly. They can be destroyed sometimes, can be affected, so there are some transformations. But basically, atmosphere is a transport medium for other environmental compartments. It connects parts of terrestrial ecosystems, different terrestrial ecosystems. It connects them with aquatic part. So what we are thinking when we start modeling on bioaerosols in the atmosphere, first, where they come from, where they end up with, and what will be the impact at the point of arrival. In the atmosphere itself, it's like just a flying like in an aircraft or in a container. Of course, they have vital role for biosphere. It's kind of trivial. They spread the organisms across the space and ecosystems. And that's actually probably the most important part of atmospheric role in bioaerosols. This is the only way to travel far and travel fast. As long as you can survive the trip, this is the way to spread the gene, to spread the species. Cycling period is much less than 1 year. Bioaerosols do not stay in the air more than a couple of weeks if they are lucky. So the adaptation mechanism to climate change is really fast. This is how they can migrate from area to area much quicker than the climate changes, for instance, or even much quicker than anthropogenic forcing changes. So it's an adaptation mechanism as well. And yeah, there are spreads of pathogens, opportunistic ones or invasive ones, plant, animals, humans. So the impact there is also important, and it is widespread once again. Bioaerosols affect our atmosphere and climate. It's not only like a passive tracer. They form cloud condensation nuclei and ice nuclei, probably the most important roles of those. They actually affect the clouds, and it's proven. And in pristine ecosystems and pristine areas, well, there are not too many of them left in the Earth, but still there are some. In these areas, the bioaerosols are probably the most important contributors to cloud processes. They help creating droplets or ice crystals, but that also means that if we create more droplets, then there is less water in each of a droplet. They become smaller size, so they affect rain as well. And the same for ice. So they do affect weather and do affect climate. And that's also very important. And from the other side, of course, the processes creating the bioaerosols, the biological process, they are sensitive to climate because plants are sensitive. So that's kind of trivial. So if we think about amount, how many of these guys down there? If we try to look in from the bird's view, I collected it from this paper, which was published not long ago, actually a bit more than 10 years already. Some adaptations were done by Fiona Thuman from MeteoSwiss. And this is the chart, which actually says that if we look into the mass part, so how many in kilograms, that's clearly pollen. We are talking about up to, let's say, 10 micrograms per cubic meter if we think about pollen season. If you think about numbers, because who cares how much mass is there, the number of particles may be important. In some cases, by far the leader is the viruses. And it's also by far the most uncertain. If we think about emission, how much of them are released, probably the most significant is fungal spores. But you see the question marks out there. It means that we have no clue. Literally, we don't know even an order of magnitude, or even plus minus couple of orders of magnitude. We still don't know. And this is the level of our knowledge. Okay, at least as it was 10 years ago. I should admit we didn't go too far from that time. A little bit better in a few places, but not tremendously large. The way to analyze bioaerosols, still very recent. The most widespread and the most regular one is the manual monitoring of particles, mostly pollen, because they are large enough so they can be seen by microscopic analysis. And the most regular one, or the most significant contribution, came from Europe. That is European Aeroallergen Network. These blue dots on this map in Europe and surroundings, that's actually sites of this network. It was started in 1974. Individual sites go back to 1960s. Something like 300 sites. They open and close comparatively quickly. In the world, you see, yeah, we have a few sites here and there. Exception is Japan, but unfortunately, this network has been closed and it doesn't exist anymore. It started in 2007, but was closed approximately 10 years later. The data from European Aeroallergen Network, they are daily, so we don't have too much information about diurnal cycle. A few sites deliver bi-hourly data, but it's much more laborious. 20, 40, 50 pollen types in some cases, but predominantly 20. In some sites, you can see some fungal spores, but they are smaller and more difficult to count. The data are not shared. The data are not available in public. The network is run privately. The problems is that, first of all, apart from pollen, we have very little. The manual technique was invented in 1940s. The paper goes back to 1951. And since then, the device remained unchanged. The principle is all the same. It's this good old Hearst type technology. I guess you know it. It's tape with a sticky layer and the fan which blows onto it. Particles get stuck on the glue, and then they are put on the microscope and counted manually. With a bit of calibration, it can be made quantitative, and that's what has been done. Uncertainties are large. And since it is so tedious, there are not too many sites which survived a long time. So when we are talking about climatological scale of analysis, we are with maybe 20 sites with the time series more than 30, 40 years long. When we think about other data, admittedly not much. There are many approaches there, the filters, impactors, impingers, whatever. Cyclones are very popular, but it all samples, one or a few samples per round. Then something has to be done with the sample, so it's once again manual analysis, and the samples changes have to be every day, so there is a lot of manual work. Cultivation, staining technologies, immunological methods, they are very tedious and as a result expensive. Regular measurements are not very active, and they are certainly slow. So as a result, for vast majority of bioaerosols, pollen is a little bit aside. Available data are not sufficient, in most cases by far not sufficient for developing mechanistic models. And that's just, to start the presentation from some bad news. For instance, this is the paper which was published in 2012, and that's one of the most extensive analysis for fungal spores. But just to draw your attention that this worldwide analysis was based on 136 samples. And in some continents, there was one sample, which was, of course, enough to make conclusions about fungal distribution over the globe. And that's more or less reflecting. And this is what, if I'm not mistaken, it was even Nature where it was published. So this is really what we know about bioaerosols. So the efforts are really necessary. The necessity of new solutions has been recognized long ago. And one of the key questions and key challenges was, first, we have to go beyond pollen. A multitude of bioaerosols is necessary. Second, we need regular monitoring. Campaigns are extremely expensive and getting more and more complicated. The key challenge is to recognize the particle type. It's not enough to measure mass. We know about organic carbons. There are thousands and thousands of stations around the world with organic mass. We need type. We need recognition. We need classification of particle types. A few years ago, about a decade ago, there was a breakthrough. The new technology came to our help. Of course, machine learning was a big part of it. First of all, just a technical task, we can automate the particle traps. We can automate impactors. It's not that much of a rocket science. Secondly, flow cytometers, that's a particle counters, the real-time particle counters, which suck the particles just while it flies through, they analyze as much as possible. They got recognition capacity, they got classifiers inside, and that was the second possibility. Third, there was another breakthrough. The third generation of DNA sequencing came to the game also less than 10 years ago. That is much cheaper and much more deep than the next generation, the second generation, and of course, much more fast than the first generation, than the Sanger method. And last but not least, the laser-based remote sensing all of a sudden got some possibilities to classify bioaerosols as well. So we got several helping hands, in most cases using quite heavy IT and machine learning and quite sophisticated technology. The difference was recognized, of course, immediately. If you look at the upper left picture, that's how you see it in the microscope on that sticky tape which aerobiologists were doing for 70 years. Now, all these stained colored particles are pollen. The black one is usually debris and sand and dust and some remnants of non-biological parts, maybe microplastic as well. But as soon as we turn to these automatic devices, things become different. For instance, this is a high-resolution microscopic image from Hund Wetzlar, the POMO device, an automated version of impactor. So basically an automated Burkhardt, but much bigger and much more complicated than that. This is holographic images of Swiss-Sense Poleno. This is the scattering and fluorescence images from Player Rapid E. This is the spectral analysis of Swiss-Sense Poleno once again, and some attempts of analyzing them with clustering analysis, for instance. So what has changed with the appearance of these new devices is the way we look at pollen and with the way we look at spores. And by the way, if I'm not mistaken, these ones actually can differentiate spores on these spectra. So we are no longer relying on our eye. We rely on machine learning after that. We rely on eye of computers. And this is example for pollen remote sensing. So the multi-band LiDAR can differentiate between some pollen families. You can see that there is dependence of this parameter, some betula versus pine. And you see that with some effort, you can argue that the number concentrations of birch is somewhere here, whereas pine goes all the way towards this curve. I'm not going into details of this, how technically it was done, but basically you can argue that there is some curve, and based on this slope, you can differentiate between pollens. Uncertainty is very large, but at least we can get some glimpse on the story. And we no longer analyze a single particle. We have to analyze a cloud of pollen. But it compensates by the fact that for the first time in history, we can see vertical profile of pollen in the air, or at least pollen families in the air. I said already several times that arguably the most complicated part is the classification algorithm, because the images are not human readable, and more information is available from each of these devices than a simple image, or by eye you can get it. I mentioned already a few. So image recognition firsthand, that came as the most straightforward idea. And all the way down to just a camera from a mobile. It actually can be used if you add a couple of lenses. It can be used as well as the cheapest versions which have been tested and still are under testing and development. There is spectral analysis if the device provides some spectra, computer vision model if scattering is involved, pattern recognition, especially if we talk about some spectral analysis, we can recognize patterns of spectra, and whatever else. More and more ideas come on the way. What is very important, and that's really a new thing, is one of these small big things when we are thinking about it. No information is given to the classifier about the particle origin, the season, when it was found, how it was obtained. It doesn't matter. What the classifier sees is just a bunch of images or spectra or some scatters. Human analysis usually they know where the tape came from, they know at what roof it was collected, and they know about the device. So they have a lot of side information which helps analyzing the particles, helps recognizing the particles, because they more or less know when a specific type of pollen or specific type of spore can appear in the air. But it also has a backfiring effect that when a new particle came, invasive particle came, climate is changing, something new appeared in the air, it might be unnoticed for years. And there are some very bizarre moments when in some countries the particles were not recognized as a correct pollen type because they thought it cannot be there. So they were attributing it in a wrong direction. But for these new machines, from the very beginning, they don't know anything about beyond the images which they have. And the goal is to get a high probability of correct classification of a single particle. Let me show you, it's actually a little bit obsolete already image, but I still like it. It's one of the first confusion matrices for Swissens Poleno. You see, these are the particles which were given, these are the particles which were claimed, and as long as you have 100% on this line, you are in perfect recognition. And this is laboratory experiment. We knew exactly what we give to the device, or as exactly as we can clean the sample. So we expected something like 99.8, which was really nice to see. But not always. In some case, you basically 50-50. You can get it right, you can get it wrong, which means that the particle is so similar to some others. You see that it was mixed with a few others or just considered as garbage and thrown out. So this is one of the first attempts which show that first, some pollens we really see way over 90% skills, which was nice. Some pollens we have problems already in laboratory. When we go to outside world, things will become really problematic. There was a campaign in 2021 which was comparing several these big devices, the new machines, and several classifiers. So how to read these charts? Each chart is one type of particle. It was in real life, and the bottom line, the perfect data were coming from Hurst type and manual analysis. There were 4 manual devices standing in different part of this pretty big roof in, it was in Germany, in Munich. So there were 4 manual devices standing, their data were averaged, and we consider that as the perfect case. So every automatic device, one color is one type of automatic device and one type of classifier. There can be several classifiers for the same device. And vertical is correlation, just temporal correlation coefficient. Horizontal is model error. So upper left corner is the perfect recognition. Correlation 1.0, model error 0. So you see that with Birch, quite a few, let's put it this way, devices were reasonable. For Fraxinus, correlation is not so perfect anymore. You see 0.5, it's already a very comparatively low result. Even worse for Grasses, for Quercus, not that bad. For the total pollen, the results look really like next to disastrous for most of the story. But since we were mostly interested about specific types of pollens, the conclusion was that, yes, several devices with several classifiers can recognize several pollen types. Not all devices, some of them were ruled out completely. Some classifiers were abandoned after this campaign. So we found specifically, I already mentioned these 2 devices, Swissens Poleno and Hund Wetzlar BA500. These 2 devices with some classifiers which I mentioned here, they are fine. And okay in this sense means that the devices were working reliably. Quite a few actually get broken during the campaign, but these 2 machines proved reliable. They showed high correlation, so they are high in these charts. And the model error was not large, and the scaling factor was close to 1, which means the device saw approximately as many pollen of specific type as Burkhardt, as Hurst device showed. It's not much use. For instance, if you miss 90% of particles, it doesn't help if you recognize correctly remaining 10%. Still not good enough. But with these 2 devices and with these 3 algorithms, it was working. So at the same time, I mentioned already DNA 3rd generation technology. We looked into this and decided that it's probably the ultimate recognition technique. Compared to the previous generation, which is called next generation in DNA world, it provides much longer DNA reads, in fact, 10 to 100 times longer, which allows a larger part of genome to be sequenced. Smaller sample needed, and it's fast, really fast. You can have 60, now the new version of the device actually allows up to 96 samples within 12 hours. The target, which we have not yet reached, I have to confess, but calculations show that once this technology is put on pipeline, you can get as low as 40 euros per sample. For comparison, this previous generation technology is about 5 to 10 times more expensive. And accuracy is pretty much okay, 97, 99%, depending on the software chemical sets. Okay, I'm a little bit pressed by time, so I show you just one example of this. I don't know, this upper line, I don't know how to get rid of it. Sorry about that. But just to tell you that this is the composition which we extracted from eDNA, collected in 25th of September in 2024, one in Lithuania, that's Šiauliai. Another one is in Novi Sad, this is Serbia. The green part is bacteria, and all red is eukaryota. So pollen spores, for instance, these are some remnants of human cells, even they are also in the air. The interesting part, what we saw here, well, probably you are not able to see, but there are some pieces of trees, and for instance, this classifier says that this is DNA from birch. But this is September, flowering is over. What happens? Our guess is that either remnants, just breakouts, garbage from trees, or seeds, which is also a possibility. And they were collected, they were sampled to a filter and then collected, we got DNA. In bacteria, a little bit scary result was that we see a lot of E. coli, Shigella, Salmonella, so quite a lot of very nasty species were found, and they were found routinely in the air. They are pretty common in the air. Concentrations are not very high, but they are there. On the left side, it's just a pipeline which was capable of creating such a picture within 12 hours. That's so far the result of the Silva project, which is developing all this technological side. So now let's jump to the model. I still have something like 10 minutes for that. If we look into model, there is plenty of options now, well, plenty. In Europe, we have two independently developed models. One is SILAM, one is CosmoArt. SILAM has a little bit more species and it's covering the whole Europe. It's now IconArt, actually. They changed meteorological driver. Cosmo is a German, developed by a consortium led by German Met Service. Now it's Icon, which is the leading meteorological model there. Art is the air quality edition, which has pollen in it. So currently there are 5 of them. There is Copernicus Atmospheric Monitoring Service, of course. That one uses 10 species, the same as SILAM, these black ones, because the source term, the phenological model for Copernicus models, was taken from SILAM. We shared our code with 10 other models. Copernicus has 11 models running, so 10 of them plus SILAM. Forecasts cover Europe. Alpine region is IconArt. Northern Europe is SILAM, Belgium, Austria. There are some local incarnations of SILAM now. Also Bavaria is added to the line. The forecasts are up to 5 days ahead, open data, open pictures. I can tell you that within a couple of months, I hope there will be a publication of World Meteorological Organization Air Quality and Climate Bulletin, where we listed much more examples of forecasts for bioaerosols worldwide. But you have to wait till late autumn when the bulletin is over, out of printing. The reanalysis has been done for Europe, and I'll show a couple of examples of that one. And okay, footprints, I probably skipped that. Thinking about technological side, how this modeling is done, I already basically mentioned that we have to know where the species are, and that means that we need quite detailed information about land use, and it's not enough to say that this is broadleaf forest. We have to say what species are there and how many classes are there and how much of each tree is per unit area per 10 kilometers squared or something like this. Then we have to have a phenological model, which is embedded in SILAM. It's driven by meteorology. It can be affected by fires, by ship traffic, by anthropogenic sources. So this like a common cocktail in this inside SILAM, and in the end we get maps of concentrations, depositions, optical thickness, and other parameters. And then of course evaluation. In a slightly different view, more oriented towards pollen, these are examples of habitation maps. Okay, this is meteorological map as another input, but if we think about birch, there is a clear peak or concentrated areas covered by birch forests. Grass is much more distributed and it's more on the south. Olives, we know that there are regions in Southern Europe, Mediterranean, in Africa, where they grow and cultivated. Ragweed is invasive species, but so far that's the area of distribution, and so on and so further. So these are the habitation maps. And this is the major and one of the most difficult prerequisites when we go into new pollen type or taking into spores. We have to say who will release them, and then we have to say when they are released. So we have to have a flowering model, the phenological part. Usually it is following the development of the season, so the heat sum is the most relevant one. And in the end, we get some nice pictures like this one. How good is the current model? That's example for comparatively recent comparison for spring in 2021. This is Helsinki. The upper chart is the result from Paleno. This is one of these automatic monitors, so very high resolution hourly data, in fact. The black one is Alnus, blue is Betula, red is Corylus. You see it down here. It's lower concentrations. And these are some false recognitions. So Paleno mixes up birch with Alnus, with Corylus a little bit. Basically, it flowers early, so this is more realistic probably. The important message is that down is SILAM, so we actually see the season, both for Alnus and for Betula, the main season, but the model tends to make it a little bit wider, and it created a much more significant aftermath of the season. You see that peak. In fact, it was greetings from northern Russia, where the model suggested much wider season. So while the blow was coming from northeast, the concentrations were considered to be high. The same happened to be in birch, where the season was predicted a bit longer. But by and large, we can get the season plus minus a few days for the most studied parts of particles. For some of them, for some others, results are better, some worse. You will see a couple of examples further. Now, what we do with a simulation, one of ways to improve the model skills, and that's what we've been discussing while preparing the cost action proposal. We can do quite a lot of assimilation if we have the data. So the model can be partly at least data driven. What I've been talking so far, the data were not used at all, only for evaluation and parameterization, of course. But if we can have some data and we have the model forecast, this blue line here, we can pull the model towards the data each time we see a new measurement coming. The model will relax back to its state of the equilibrium state. So we will get some these relaxation curves, which are shown here. And when we unify them into a single forecast, we get something like a saw or a hairbrush. And that's actually how the weather forecasts are done all the way now. And if you look carefully into weather forecast, you will see that red line with broken moments when the data assimilation happens. This is one of ways. Another way, and that's what we use mostly, is the source term inversion. If we see the same run, we run the model, and we see concentrations. Now we observe concentrations. Then we get the first measurement. We go back in time, and we figure out which source it was responsible for that particular measurement. We can do that because we have the model. We go back and forth in time. And then we change the source so that the model result is different. So it now passes through the first dot. Then we get the next dot. We go back again. We find the source responsible for it and adjust the model. And then we continue all the way. And in the end, we get this thick red curve, which is closer to measurements. It's not close to any specific measurement. You can compare it with this hairbrush style of baseline assimilation and choose which one you like more. But I should say that much more reliable is the source term inversion than this forecast with baseline basic assimilation. Okay, I think I skipped the data fusion part for the sake of time and go to the last topic of my presentation. It's already almost 40 minutes were spent. This is the European pollen reanalysis, which was built just long ago, not long ago. It says in press here, sorry about that. It's actually published already. This is the DOI for the paper. It was published late in 2024. For 3 species, alder, birch, and olive, for 43 years, CLAM was run for the whole Europe, and we used the pollen observations, which I described, the manual observations for these long times from 34 European countries. So that's how on the right side, you see this total pollen load, just integral of concentrations, averaged over these 43 years for the 3 types of pollen there. I think I skipped these ones, maybe spent a minute on that chart. The important message for this pollen reanalysis, yes, we used source inversion firsthand. We used 4-dimensional variational data assimilation. For those who use this method, you know what I'm talking about, but I'm not going into any details here. But in the end, we get the assimilated run where we adjusted the source terms and then let the model run freely. But the feature of this selection of the source inversion with use of 4D-var, but actually with any other method of data assimilation, is that you will get biased solution. So there had to be a bias reduction and some recalibration of the system afterwards. As a result, we get the pollen reanalysis, and that's an important part. Okay, I skipped this once again. There was some comparison on that. When we were thinking what we assimilate, I said that we assimilate pollen features of the source. And we decided that the most uncertain part is the total seasonal release per meter square of the forest. Because we don't know how productive trees that particular year, and it's extremely difficult to predict. So we assimilated it. The result is actually quite spectacular. You could see the distribution of correlation coefficient. The more to the right, the better. This bluish bars are for the reference run. This correlation between the years, alder, birch, and olives. And you can see that there is quite a lot of stations which said that the correlation is very low. So it's even negative in this particular case. After assimilation, we get a much higher correlation, much more stations with very high correlation. And the same was for birch, a bit less trivial for olives, but there the situation is really difficult because the plant is human controlled. We cannot really rely on environmental parameters alone to simulate it. So it's more difficult in that sense. In time series wise, it looked like this. Here it is for Stockholm and for Turku, so Sweden and southwestern Finland. This dashed line is just seasonal pollen load year by year from 1980 to 2022, you see that the model overestimates, red line is observed values. So model overestimates and overestimates quite a lot, actually, factor of times. Then there is some very high year when we underestimate, or another year when we once again were underestimating. The blue is after assimilation. So you see that it was possible with assimilation to bring the model really close to the measurements. Not in the peak, I promised you some underestimation, here it is. The same also here, so peaks were underestimated, we had to recalibrate the model. Okay, I think this is my conclusion. So the challenge for the future, and that's my conclusion, that we need to bring together the in situ measurements. I showed you a little bit about them. I showed you a little bit about LiDARs. I showed you a little bit about DNA. They are independent. They talk about different features. They rely on different methodologies and they are complementary. And the models, they also rely on completely different features and they are driven by different processes. We are starting to bring them together. We need machine learning to do that. It's a very multidimensional problem. But that's probably the way to go with our current, what we have at hand now. Thank you.
Thank you so much, Mikhail. It was really interesting for me, especially the data assimilation part. Thank you so much.
Welcome.
Are there any questions from the audience? Yeah, Istam, go ahead, please. Istam, we cannot hear you. It's muted.
Okay, sorry. Yes. Hi. Hi, Mikhail. It was a great presentation. Thank you so much. I don't know about the others, but it was very clear for me. And I have one question about this. So you mentioned that you have several breakthroughs with data. I was wondering if any of these led to some breakthroughs in the modeling as well, or are you just harvesting these benefits through data assimilation only at the moment?
Actually, this is this slide, which I skipped. The breakthrough in the data, it's firsthand, it's data availability, and second, it's data timing. Data availability is just political decision. When we started this automatic networks, it was a principal decision that this data will be wide open from the very second they are obtained. And that's a totally different approach. There are still debates whether it's a good idea, but the decision has been taken. And the timeliness is the second thing. What is here is the forecasts one by one. It is in Bavaria, in Munich, I believe, just one station. Blue is the measurements. And the orange is forecast day by day. So we go one day, each day next data come, we go one forecast one day next. And we use the information from previous days. And you can see how the assimilation of this real-time data adjusts the model. And in the end, when we come to like middle of the season, the model already adjusts its strength, adjusts its timing. You see that some false peaks which were at the beginning, they disappear very quickly. So this is what we get out of the data availability. So in that sense, you are right. The biggest impact, what we expect from these breakthroughs in the data, is the data assimilation. And I should say that this technology which was developed, it's by itself a breakthrough. We didn't know how to assimilate pollen because we never had the data and we never had them in real time. So we simply didn't know how to assimilate it. We knew it. It was actually a game with Silam. I published it in Aerobiology maybe 5 years ago or something like this. When I showed that meteorological type of assimilation, this hairbrush, will not work. The assimilation effect will be forgotten within a few hours, something like 5 to 6 hours maximum. They are forgotten completely. And the data are daily, so how to assimilate daily data if you forget them within 5 hours. That has been shown, but we didn't know what to do. So this is actually, I would say, the biggest breakthrough with the modeling side.
Are there other questions? Okay, Nigel, go ahead.
Yeah, thanks. I agree with Fair that the presentation was really very clear. I was just kind of curious about the use of eDNA. Do you have really kind of you are able to use eDNA in a so short time scale, that is, you get the sample and you can analyze it and get the results within a few hours, one day, or?
That's real.
Yes.
So basically, what we do now, we do not target 12 hours. I should admit, we target 3 days. So what is actually happening? The filters, if they are collected, let's say at the FMI roof, we have the filter by the evening. Overnight, sequencing goes through, and we need a few hours for the recognition algorithm for classifier, which will go to GenBank and other DNA libraries with recognition and recognizes it. So basically within something like 12 hours, we can get that thing running. There is a contradiction there, I should confess it. Analyzing one filter with this third generation will make it astronomically expensive. So what we actually do instead, we collect filters from several locations. We wait until we have now it's 24 at least or 96 filters to analyze. And then they are put into pipeline because the device, it's nanopore, Oxford nanopore, GridION and ProMetION. We have this one cell from ProMetION actually, which can do 96 samples at a time. And then we put them all 96 together, and then it's 12 hours, and we have almost 100 samples processed. With the pipeline, it goes sample by sample, but it can be parallelized because this is just computer exercise. So yeah, we actually can do it within this time.
Yeah, well, that's impressive. Thanks.
Thank you.
Any other questions? I have one in the meantime. I think one of the barriers behind not being able to do near-term forecasts is also the data availability, as you said, but also the fact that data is publicly available. And you said for the European network that you had, the data, for example, was not shared. So I was wondering, for the pollen data, what are the conflicts of the data not being publicly available?
Well,
maybe it's a too complicated question.
Well, every complicated question can be answered shortly. In short, history. The driving force for making that whole thing running in middle of previous century in 1950s was allergy. So the first stations were run not by scientists, they were run by doctors. They were initiated by hospitals. And the hospital wanted to know the situation on their roof, and they didn't care about anything else. So it's history. They later came to a network. Then they figured out that it is expensive and boring to do this analysis. So it started becoming also a commercial side. Then they figured out that there are companies like I don't know, Bayer or some others, allergy pharma or whatever. So big pharmas who develop antihistamines, for instance, or some anti-flu or whatever drugs, they have to test them. And they testing against pollen, you have to know when pollen is the air. So these big companies started buying this data. And they were targeted from the very beginning that, if you have a drug which was costing development something like a couple of billions, for instance, and the test of it costs 10 to 100 million euros, okay, you can afford 50,000 for pollen data. So this is how the network was born. things do not get easy with years, with the network growing and the income shrinking. So there were countries even which were losing the network. But this initial construction as a collection of privately run individual devices, it remains still today. And basically for this reanalysis, we literally had to go station by station, agreeing with every owner of the trap that they allow to use their data. So that's really how it was done. In a few cases, it was national network. That's true. Like in France, for instance, was national network. The same in Finland. So we were able to go country by country in a few cases. But in quite a few cases, it was literally going trap by trap. So that's just history.
Yeah. Thank you. Are there other questions? Yeah, Istam, go ahead.
Just to follow up to this one. So in the eDNA results, for example, you saw now that there's also bacteria and other unexpected things are in abundance. Does this added value kind of will benefit pollen research because maybe others now will also start taking measurements because they have other things that they are interested in or not?
Yeah, well, a simple answer is yes, of course. A more complicated one, I should say that this eDNA part appeared to be, first, tremendously more complicated than we thought for many reasons. And second, it brought us so many surprises that I don't even know which of them is just an artifact and which of them a breakthrough. The thing is that Illumina, this next generation sequencer, and PacBio, they are so popular and they are the only ones in the market. Okay, no, very few people actually use Zanger methodology of this first sequencing. It's too slow, too expensive, but they are so popular, they're practically the only ones, that all software for bioinformatics, for instance, it's all Illumina. When we came with Nanopore, yeah, sorry, this software just breaks down when we give it Nanopore sequences. They are used to 200 base pairs, what Illumina gives. When we give them 6,000 base pairs read, it just overfills the memory and breaks down. So instead of just plugging it and starting working, we had to redo lots and lots and lots and lots of things, just recreating the pipeline for bioinformatics, which is existing for Illumina. We had to make it for Nanopore. Some software was used, some software was created or adapted. So that's really complicated side. And well, maybe one example, we were really surprised to see some eagle from South America. We saw it in Helsinki. DNA. How comes? It appeared to be artifact. Once we corrected some biases in the software, yeah, it recognized correctly because you still don't have the full genome even from Nanopore. You have to guess. So, but I would say that we are still at the beginning of recognizing how huge things come with this DNA. This is really was leap in the dark. I'm not sorry by any moment that we made it, but I should say that we are well behind the time schedule which we had at the beginning, and we found much more really intriguing things which we did not expect the slightest bit to find.
Yeah, Maria, go ahead.
Thank you. Yes, thank you very much for this wonderful talk. It is so fascinating. And so, yeah, it just, I suppose it's just to admire how much effort goes into putting all of this together. And my question then is, you said it in several instances, I think you mentioned it at least, but this must be a very interdisciplinary type of work. So what is it? Like, what kind of types of researchers do you have to get involved? You know, because I suppose you need decision scientists, you need computer scientists, you need people that know I mean, who do you like work regularly with to make this happen?
You're right. Let me start from myself. I'm an engineer. Yeah, I came from computing part and radio technology part even. And I'm a computer guy. So I moved to measurements simply because we recognize that apart from amazing technological solutions, engineering solutions, we have this machine learning part, which we are the only ones who can do it. So you have to start from somewhere. If you start from biological part, who know how the particles look like, they get stuck at once. They don't have skills necessary for that. On a computing science, we know what to do with these parts, but we don't know what that means. So absolutely, we had to learn and work with many institutes and organizations. Now, some members of our team, I saw Yulia is in this telecon. She went through basic course of aerobiology, just to know how these guys look like. Just know how Hearst works, to learn how to make that tape and how to make it running. So we had to learn lots of things. And yes, we have now microbiologists in the team working for it. FMI actually established first microbiological laboratory worldwide in med services. We are the first med service who has it. And certainly we are the first ones who has microbiologists in the staff. So yes, you're right. And we work with many more people. So yeah. Our side is computing part, which for many looks like the most, at least cumbersome, if not complicated, which may be true, maybe not, I don't know. I should say that when these guys start talking biological terms, I still get lost.
No, yeah, thank you. That's fascinating. Yes, really fascinating.
Well, thank you so much, Mikhail. I think we are already we finished our time, but I echo all the beautiful comments. It was really inspiring for me to see all this work.